In February 1954, a shipment of oysters left the coast of Connecticut in a light-proof container. Packed into damp peat moss, they traveled by railway express to Evanston, Illinois, hundreds of miles from the tides of Long Island Sound. Their destination was Frank Brown's laboratory at Northwestern University. Brown selected 12, attached recording devices to the shells, and watched them for the next 46 days. Even without the ocean, the shells continued opening around the times when the tide would have been high back in Connecticut. Then about 2 weeks later, the timing changed. Brown stated posed a mystery that biology still has not completely solved today. How can living tissue keep time when the clock outside it disappears? The oysters were dredged near New Haven, held at a federal laboratory in Milford, and shipped west in darkness. In Evanston, a thread connected each shell to a spring recorder, turning every opening and closing into ink marks on a rotating drum. This apparatus produced a simple behavioral measure, minutes open per oyster during each hour. Brown divided the collected data into 3 blocks of 15 days. During the first block, the activity peaks corresponded roughly with the high tide times at the Connecticut collection site. In the later blocks, Brown reported that the peaks had shifted by about 3 hours. They now occurred near the times when the moon crossed above or below the local meridian at Evanston. They knew where the ocean should have been. But Evanston has no marine tide. Brown measured a phase relationship between valve activity and local lunar transit timing. His experiment revealed no coordinate sensor and left the cause unresolved. A schedule driven entirely by rising water should fade after the tide disappears. The continued rhythm observed by Brown suggested a process inside the animal that could run on its own. Imagine one point moving around a circle. Each turn is an internal cycle, and its position is the oscillator's face, where the system sits in its repeating pattern. The observed rhythm is pretty much free running. Its period may nearly match the environmental cycle and slowly drift. If a repeated signal pushes the phase earlier or later until the cycles synchronize, that process is called entrainment. In this model, the next phase equals the current phase plus the oscillator's own advance plus any correction from the external cue. The same model produces persistence in phase shifts while leaving the source of that correction unknown. Light, temperature, water movement, pressure, and other periodic signals can entrain rhythms in different organisms. Brown reduced some possibilities without isolating a cue that explained the Evanston shift. His study also only involves 12 animal samples in one very historical setup. A stronger modern test would use independent groups, monitor candidate cues, deliberately shift them, and disrupt the proposed sensor or clock pathway. Brown documented a persistent pattern and reported a remarkable phase change. However, his apparatus supplied no evidence for longitude sensing, tidal calculation, or a specific external force. Decades later, Damien Tran and colleagues fitted Pacific oysters with electronic valvometers in Arcachon Bay, France. 15 oysters recorded in the field opened in a strongly tidal pattern. They then put a second group in a constantly dark environment in the laboratory for 7 days. Most still remain rhythmic, dominated by approximately tidal periods. These were Pacific oysters, a different species from Brown's original eastern oysters. Researchers further discovered that some familiar circadian clock genes also varied at tidal frequency. One molecular clock might respond differently to daily and tidal inputs, separate clocks might interact, and different species use different clocks. From classical conditioning experiments, we learned that mammalian brains judge whether a sound lasted half a second, learn when a reward should arrive, and release movements at the right moment. These shorter intervals recruit neural circuits with very different biology than what we see in oysters. That connection is computational and not purely chemical. In 2004, psychologists Matthew Mattel and Warren Mack developed a corticostriatal version of the beat frequency model. Their computer simulations treated the start of an interval as a reset for neural oscillations running at different frequencies, so the combined phases continuously changed. Picture 6 hands rotating at different speeds. Each moment produces a new arrangement. At a rewarded target time, their joint pattern becomes a neural barcode. Mattel and Mack modeled striatal neurons learning this barcode. Reward strengthened the inputs active at the target moment. Later, a similar phase combination activated the detector and signaled that the interval had elapsed. Adding noise and the dynamic response threshold reproduced patterns from peak interval tasks, where animals learn when the reward usually arrives and keep responding during unrewarded probe trials. The combination carries the interval, and each oscillator supplies one part of the pattern. This then creates a pattern encoding problem. Too few oscillators make different moments look alike. Noise prevents perfect matches. Frequency drift moves the response early or late. The code must therefore distinguish nearby moments while tolerating biological variability. 6 perfect sine waves are an illustration of coordinated corticostriatal activity. Mattel and Mack's proposal connects timing behavior with plausible circuitry. This theory was carried forward practically in 2015 when Thiago Gouveia and colleagues at the Champlimod Center in Lisbon trained rats to categorize intervals around 1.5 seconds. In their experiment, a nose poke triggered 2 brief tones separated by a variable pause. After the second tone, the rat chose a left or right port and received water from the correct long or short judgment. Electrodes recorded groups of striatal neurons throughout the interval. Their population activity followed an evolving trajectory. Faster trajectories accompanied long judgments. Slower trajectories accompanied short judgments. Infusing the striatum with mucimol reduced sensitivity to duration. That experiment tied durational judgments to a change in neural population. Its design could not determine whether beat frequencies generated that trajectory. During the quiet 4 period before a task stimulus, they measured how quickly fluctuations in firing lost their correlation. Sensory areas showed shorter timescales, while association and prefrontal high-performing areas carried activity across longer windows. Shorter windows can follow rapid sensory change, while longer windows can integrate information or maintain context. The Oyster oscillator, striatal phase detector, population trajectory, and intrinsic neural timescale hierarchy describe separate mechanisms and measurements across very different scales. They share an abstraction. A changing state can make time inferable. Another process can read that state, compare it with memory, or adjust it using environmental signals. Brown's oysters carried no map of Illinois. Their tissues generated a changing state that Brown could record. Similarly, neural populations generate changing states that downstream circuits can read. One marks the return of an absent tide, another can mark a vanished clock. Brown pursued biological rhythms for the rest of his life, even as chronobiology moved decisively toward endogenous clocks and later their biochemical and genetic machinery. He and his colleagues tested whether magnetic fields, weak gamma radiation, and other subtle geophysical signals could influence biological timing. Those interpretations remain controversial. They did not overturn the endogenous clock model, and they do not explain the oyster's phase shift in Evanston. But the question at the center of Brown's work survived the dispute. An organism can generate a rhythm internally and still use signals from the outside world to set its phase. The signals and sensors differ across species, and the oysters remain unresolved. Brown died in 1983 without settling what moved these activity peaks by 3 hours. He left behind a harder version of the mystery, not whether life keeps time, but how an internal rhythm becomes aligned with the changing world. And all of this began with 12 oysters that seemed to track lunar time.